Hello everyone, welcome back to the channel. Today we are diving into the microbiology laboratory to identify MRSA, which stands for Methicillin resistant Staphylococcus aureus. We'll walk you through sample collection, culturing, biochemical tests, antibiotic resistance, and infection prevention. Whether you're a student, a laboratory technologist, a laboratory scientist, or a biomedical scientist, or you are just curious about microbiology, this will provide a step-by-step guide into identification of MRSA in a clinical microbiology laboratory. Staphylococcus aureus is a gram-positive bacterium which is a cocci in shape and lives on our skin. For most people, it causes no harm, but if it enters the body, it can cause infections ranging from skin abscess, pneumonia, sepsis, endocarditis, and other infections. Originally, Staphylococcus aureus was treatable with the antibiotic penicillin, but resistance quickly developed. In the 1960s, methicillin was introduced as a solution, but soon after, resistant strains of Staphylococcus aureus called methicillin-resistant Staphylococcus aureus appeared, hence the name MRSA, which stands for Methicillin-resistant Staphylococcus aureus. MRSA carries the gene MAC-A and MAC-C, which produce an altered penicillin-binding protein called penicillin-binding protein 2A. This protein blocks nearly all beta-lactams antibiotics from working, making MRSAs a serious therapeutic challenge. MRSAs can be acquired from the hospital called healthcare-associated infections or from the community. MRSAs is the leading cause of hospital-acquired infections worldwide and continues to spread globally, making its detection and control critical. Sample collection. Sampling can be done from wounds, surgical site, the nose, the axilla, the groin, or other body fluids. Diagnosis begins with a proper sample collection, which could be swabs from the nose, from the groin, or urine for screening purposes. Sample transportation to the clinical laboratory. Samples are transported to the clinical laboratory in a temperature-controlled environment. The transport medium is usually a maize medium in the form of charcoal or gel swabs. To culture in the clinical laboratory, first, you have to ensure that the patient's identification on the swab matches the identification on the form, after which you have to register in the clinical laboratory information management system called LIMS. Specimens are plated onto selective and differential chromogenic media. Specimens can also be plated onto blood agar, or they can be plated onto mannitol salt agar. After specimens are plated onto agar plates, they are then incubated at 37 degrees Celsius for at least 18 hours. If there is growth on any of the agar plate, it has to be identified first before further testing. Preliminary identification of Staphylococcus aureus. First, we'll look at the catalase test. All species in the group Staphylococci are catalase positive, which means that they are able to reduce hydrogen peroxide into oxygen and water. The released oxygen produces bubbles, which is a positive catalase test. Coagulase test. Only Staphylococcus aureus in the group Staphylococci is able to produce the enzyme coagulase that leads to clotting of plasma. In order to confirm the presence of Staphylococcus aureus, you can use either a DNA hydrolysis test, which in this test, Staphylococcus aureus produces a zone around itself upon addition of hydrochloric acid in the DNA's agar. You can also confirm Staphylococcus aureus using MALDI-TOF mass spectrometry. MALDI-TOF stands for Matrix Assisted Laser Desorption Ionization Time of Flight, or you can use Vitek2 identification system. After confirming the presence of Staphylococcus aureus on the sample, to confirm the presence of MRSAs on the sample which you have collected from the patient, Antibiotics are usually tested in a clinical laboratory in order to determine resistance to methicillin. Commonly, oxacillin and cefoxitin are the antibiotics that are used in the clinical laboratory. Cefoxitin is commonly tested because it is more reliable and it activates the MRSA gene. Therefore, to confirm Staphylococcus aureus as being methicillin resistant, it has to be resistant to either oxacillin or cefoxitin. You can also confirm the presence of MRSAs using a rapid immunoassay. Confirming MRSAs can also be done by a molecular technique such as PCR to determine the presence of MRSA and or MRSA genes. In clinical practice, MRSAs is both a scientific and a clinical challenge. The bacterium adapts rapidly and resists many of the commonly used antibiotics. Therefore, accurate testing and strict infection control is required to keep the spread of MRSAs under control. Prevention of MRSAs. In order to prevent the spread of MRSAs, strict infection control measures must be maintained. This could include proper hand hygiene with soap and water or alcohol-based hand rub, effective triage and screening at the hospital or at admission, decolonization protocols such as nasal ointment for carriers. Methicillin-resistant Staphylococcus aureus can also be prevented by taking the right antibiotics for the right duration.